everyone, welcome to Biomixer. Hope you're doing well. So in the last class we have discussed how the link reaction is linking the glycolysis and the Krebs cycle. So in this video we will discuss about the Krebs cycle or the citric acid cycle. So what happens in case of the link reaction? What we got the acetyl coenzyme A. So in the mitochondria, the Krebs cycle which is taking place, where it is taking place? It is taking place in the mitochondria and specifically in the matrix. Okay, so due to the link reaction, what we got the acetyl coenzyme A. Here two molecules of acetyl coenzyme A is being taken through the link reaction. So in the matrix, what happens? There is presence of oxaloacetate. Oxaloacetate is present in the matrix. It is known as oxaloacetate, and this oxaloacetate which is present, it is a 4-carbon containing compound. So the acetyl coenzyme A, which has been taken by this link reaction inside this mitochondrial matrix, it is being used here. Acetyl coenzyme A, which is produced, it is a 2-carbon containing compound. So here it is being utilized. 4 plus 2 equals to 6. 6-carbon containing compound is formed, which is known as the citrate, and this is the first stable compound. That is the reason, first product, that is the reason it is known as citric acid cycle. So during this case, what happens? There is requirement of some of the enzyme. So what is the enzyme? It is the citrate. It is the citrate synthase. Okay, it is the citrate synthase. And when this acetyl coenzyme A is used, during this time there is removal of this coenzyme A. Only the acetyl group or the 2-carbon containing compound is being utilized in this case. Okay, now this citrate which is present, it is a 6-carbon containing compound. So this citrate, it is being converted into isocitrate, and there is an intermediate during this conversion of this isocitrate. Here what happens during the formation of this isocitrate? There is removal of water. H2O is removed, and this is a reversible phase. Okay, here there is removal of water, and again there is addition of water for this step to take place, and in between there is another one product which is formed, which is known as cis aconitase. Okay, so this enzyme, the enzyme which is required in this case, it is the aconitase. Okay, it is known as aconitate. Okay, so the next step is from the conversion of isocitrate into alpha ketoglutarate. From this isocitrate which is present, it is also a 6-carbon containing compound. So after this isocitrate, it is being converted into alpha ketoglutarate. During the conversion of isocitrate into alpha ketoglutarate, what happens? There is removal of CO2. That means now a 5-carbon containing compound is being formed, and NAD plus which is present, it is being converted into NAD and NADH and H plus. So for this, what is required? It requires one of the enzymes. So what is the enzyme? It is known as the isocitrate dehydrogenase. Okay, so this process which is taking place, it is known as the oxidative and decarboxylation phase, because here the carbon dioxide is being removed. That is the reason it is known as decarboxylation, and the NAD plus is oxidized into NADH and H plus. That is the reason it is known as oxidation. This phase, this third step which is present, this is the third step. This third step is known as oxidative decarboxylation phase. Okay, now this alpha ketoglutarate which is present, it will be converted into succinyl coenzyme A. Okay, here it will be converted into succinyl coenzyme A. So what happens in this case? This is a 5-carbon containing compound, but the succinyl coenzyme A which is present, it is a 4-carbon containing compound. So in this case, there will be release of one of the CO2 molecule. One carbon dioxide will be released, and NAD plus will be converted into NADH, and there will be utilization of coenzyme A. Okay, so this is the fourth step. So here what is the requirement of the enzyme? It is known as the enzyme which is responsible is alpha ketoglutarate dehydrogenase. So this phase, what it is, how the reaction is taking place, it is taking place by the decarboxylation as well as oxidation. Okay, so here it is known as oxidative decarboxylation. So here there is formation of succinyl coenzyme A. This was the fourth step. So in the fifth step, what happened? It is forming the succinate. It is the fifth step. In the fifth step, what happens? There is removal of this coenzyme A. Okay, so during this time, when there is removal of this coenzyme A, here the ATP or GTP is also taking place. So here there is requirement of the coenzyme A in the previous step, then the removal of coenzyme is taking place. So why it is so? Because by taking the coenzyme A, it is forming the succinyl coenzyme A. And when the succinyl coenzyme A is divided into two parts, that is the succinate and the coenzyme A, during this time there is formation of the energy. Here the energy can be produced. That is the reason first addition is taking place and the removal is taking place. Okay. So in this case, there is requirement of the enzyme. What is the enzyme? It is known as the succinate. It is known as succinate thiokinase. Kinase, that means it will give some phosphate, and the phosphate will be taken up by the ADP or the GDP to form the ATP or GTP. Okay, now next, the succinate which is formed, it is also a 4-carbon containing compound. The succinate, it will form the fumarate. Okay, this fumarate which is produced, it is also a 4-carbon containing compound, and the formation of succinyl coenzyme A to succinate, it is also a reversible phase. Okay, during the conversion of succinate into fumarate, here FAD is converted into FADH2. So here the enzyme which is responsible is succinate. It is succinate dehydrogenase. Okay, now the seventh step is the conversion of this fumarate into malate. This is also a reversible phase. Here the malate is being formed, which is a 4-carbon containing compound. So here the enzyme which is responsible is, it is the seventh step. Here fumarase is being used. So next step is the conversion of malate into oxalate. This is also a reversible phase. During this time, the NAD plus is converted into NADH and H plus. So the step which is present, it is here the enzyme which is required is malate dehydrogenase. Okay, so here in this case, from where the succinyl coenzyme A is converted into succinate, during this time, this step is known as substrate level phosphorylation, because here the coenzyme A is removed in this case, and there is also formation of this ATP or GTP. That means the phosphorylation of the ADP or the GDP is taking place along with the removal of this coenzyme A. That is the reason it is known as substrate level phosphorylation. Okay, so you have to remember what are the enzymes required, what are the 8 steps required in this Krebs cycle, what are the enzymes required, what are the end products they are getting, like how the after the decarboxylation or after the oxidation, what is taking place, how many NAD plus, how many FADH, and how many GTP are being produced. So here you can see here that is removal of one of the NADH over here. The second NADH is here, so 2 NADH, so 3 NADH. So here in total we got 3 NADH, 1 one FADH2 and 1 GTP. Okay, when we use 1 molecule of acetyl coenzyme A. So so we got how many molecule of acetyl coenzyme A? We got 2 molecule of acetyl coenzyme A from pyruvate. So everything will be multiplied by 2. So in total, how much NADH we got? In total, we got 6 NADH, 2 FADH2, 2 FADH2, 2 ATP or GTP, which we have got from this Krebs cycle. Okay, so let us see what are the steps which we have followed during this Krebs cycle. So at first, in the first step, so there was 8 steps in this Krebs cycle. So in the first step there was condensation. In the second step there was dehydration and hydration. In the third step there was oxidative decarboxylation or dehydrogenation. In the fourth step there was the same oxidative decarboxylation and dehydration. In the fifth step there was phosphorylation. In the sixth step it was having dehydrogenation. In the seventh step it was having hydration. And the eighth step, it was having dehydrogenation. So these are some of the steps which was followed during this eight phases, or the eight phases. The acetyl coenzyme A is taking place in the 8 steps. So these are the steps which are used in this case. Okay, so here what we at the end what we got 6 molecules of NADH, 2 molecules of FADH2 and 2 molecules of ATP or 2 molecules of GTP. So we will calculate here the overall what are the things which we got through the process of glycolysis, link reaction and the Krebs cycle. So when we use 1 molecule of glucose, when 1 molecule of glucose was used and there was formation of 2 molecules of pyruvate. During this time what we got 2 molecules of NADH and 2 molecules of ATP. So here how many ATP is produced? When we say here 2 into 2.5, that means 5 ATP plus 2 ATP. So in total how much ATP? In total we got 7 ATP in this case. Next step was the link reaction. During this link reaction what we got? Link reaction, it was forming the 2-acetyl coenzyme A. During this time we got 2 molecules of NADH, that means 2 into 2.5, that means 5 ATP. Okay, next in the Krebs cycle what are the things which we got? We got 2 FADH2, 2 ATP and 6 NADH. Okay, so here what we can get? 6 into 2.5, okay, plus 2 into 1.5, that is for the FADH2, got 2 molecules of ATP. So in total 15 plus 3 plus 2 equals to 20 ATP. Okay, 20 plus 7 plus 5, 27, 28, 29, 30, 32. So in total we got 32 molecules of ATP during the conversion of 1 molecule of glucose to pyruvate to acetyl coenzyme A, and from acetyl coenzyme A into the Krebs cycle. So in total what we got? We got 32 molecules of ATP. Okay, so here what happens? So here we can see here the 32 molecules of ATP is formed from 1 molecules of NADH when we are calculating in the form of 2.5. But if we calculate in the form of 3, approximately 3, during this time what you will get? You will get 38 ATP. Okay, when the calculation will be done for NADH, what we are taking? We are taking 2.5 molecules of ATP. But if we take 3 molecules, approximately 3 molecules of ATP, and for FADH2 we will take approximately 2. During this time we will get 38 molecules of ATP which will be formed. So in the questions also you will be seeing that. So here what we saw? We saw that there are 32 molecules or 38 molecules, but in the question you can see there are 30 as well as 36 molecules. How does 38 is being converted into 36 and 32 is converted into 30? Okay, it is due to the subtle system. Okay, so in the next class we will discuss how the subtle system is taking place.